back a little bit. So these are those elements that are kind of far away from the start site, but they regulate gene transcription. So this is a really cool picture that shows, more detail on how these enhancers might be working. So this is a neat thing because, we all know that the DNA is really long, right? There's a lot of information there. So the DNA looping is a structure that can be formed so that we can really bring these far away enhancer sequences to the area of the promoter. So notice a couple of things in this image that I want to highlight here. We've got this cohesin rubber band thing that goes around the whole thing. I should back up. So here's our promoter, and we've learned about that C-terminal domain. We've learned about the TF2s. Oh, it's so funny. I have to share this with you. So we were in my senior level class yesterday, and we're brainstorming experiments and stuff like that, and we're talking about different assays for to develop for their project because they're going to test a drug on their cells here the rest of the semester, and then come up with assays for looking at the unseeable, right? And I was like, one really hard thing to talk about is transcription regulation. So we started talking about assays and brainstorming assays for that. And I go, you remember those TF2s and the things that you guys all have in your head? And somebody was just like, and there were like so many people that were like, oh, that's so awful. I was like, well, you better remember that because I'm making you all, this class, all remember it. But it was funny. It was just hilarious. So hopefully you don't feel that way. But if you do, I totally understand. Anyway, there's the promoter and those transcription factors. It looks a lot like a pull two setup here, or polymerase 2. So we've got that in purple, and then you've got the transcription factors in this orange and the green, and then the big mediator complex that gives the whole thing a hug, kind of locks it all in. But then this is cool. These are what the new things are. We've got these other proteins that are coming around to even add to it. So it's just like so many pieces that come together to solidify the binding of this polymerase and regulate it, right? But the cohesin is one we haven't really, I don't think, talked about yet. But cohesin you learned about when you're talking about mitosis in like earlier classes, right? Cohesin is what's holding the the sister chromatids together. Same protein, but in this situation, it's letting this nice long DNA loop come together, and it's basically solidifying or securing the enhancer sequence so that it's just in the right area, right enough, just the right enough space that the enhancer protein can bind it. Okay, so really interesting. Check my notes really quick. So just another thing to remind you about is enhancer sequences account for 10% or more of the human genome. So 10% of the genome are enhancer sequences. So just even more significance for this, there are so many disease-associated mutations of enhancer sequences. There are quite a few. So they are not junk DNA like people used to say, right? They are very important to the overall health of our cells and everything. All right, so enhancers do like so it's not that you have like the whole chromosome and any enhancer is going to help with any promoter. There are specific enhancers that are dedicated to a specific region. Does that make sense? So like it might be far away, but there is like a cassette that belongs to each other. So that's what we mean by enhancers stay in their lane. We have insulators, and these are things that are going to divide the chromosome into these domains. So I like to think of the enhancer and the promoter and the genes that belong to that. These are like a cassette or a set of genes that belong together. Or I shouldn't say genes. These are a set of DNA elements that belong together. All right, so the genome can be divided into these discrete chromosomal domains called topologically associating domains, or tags. It's always fun to abbreviate everything, but the topologically associated domains. All right, so the enhancers and the promoters that belong to each other, they exist within the tag. That's what that means. They stick together, that cassette. All right, the insulator of the TAD, sorry, or the, are we okay? It scared me like an alarm. No worries. Okay, so the TADs are like the margins of them are basically defined by the CTCF, and that's a protein. That's a protein that actually binds what is called it's calling out the type of the DNA sequence that it recognizes. And so these are the DNA sequences 3C, so it's CCCTC. So it's a CCCTC binding factor. So that's the sequence that it recognizes. So here is a nice a TAD, a topologically associated domain. It's kind of turned up on its side as you know, relative to what we just saw. So we've got our promoter, we've got our enhancer, we've got all this other sequence. There might be other promoters in here and other genes in here too, right? That would be stuck, that would be regulated by the enhancer. But then we've got these, the little yellow circles are the CTCFs, and here's that cohesin, just kind of, you know, it's like a rubber band, and it's just kind of keeping things together. But then you have the CTCFs that basically they are another block or another margin defining binding element. Okay. And then we have these cool, even more robust things around it as well. So you've got TF3C, CTCF, and cohesin, all of that stuff. And we've got like a duplicate and a duplicate on each side. So how do you find sequences that are bound by this stuff? Like, you don't know. Maybe it's not in the database yet. How do we figure this out? So the CHIP assay, or this assay that's shown here, chromatin and immunoprecipitation. Chromatin, remember, is protein and DNA, right? So this type of immunoprecipitation assay is one where you start off by pulling down the protein, right? And then you look at what is the DNA that's associated with the protein. So let me walk through it and make it a little more sense. So here is our cell, and we want to know, we've identified a new protein, or maybe we've treated the cell, and we want to know, like, okay, under this treatment with tea tree oil, is there going to be enhancer bound by some protein or something like that? So we know the protein. So what we do is you treat the cells with formaldehyde. Formaldehyde is really cool because it'll cause every protein to be cross-linked by an aldehyde group. So it basically glues those molecular structures together. So they stay stuck together. And then you can fragment the chromatin, so isolate the DNA. You've probably done this before, like in the spin columns that you've used before to capture the DNA. You can do that. You can be bound by proteins, or you can use an some sort of chemical method too. And then what you do from your lysate is, or from your DNA solution, is you can immunoprecipitate with the specific antibody for the specific protein. So here we're going after the green protein. And once we have, washed away all the other non-green proteins, then you can reverse the cross-link. So every chemical reaction is reversible. Remember that, right? So we can reverse the formaldehyde reaction to release the release the nucleic acid from the protein. And then you can just use PCR to identify, or DNA sequencing to identify what it is, right? So that's a neat thing. I think I don't know that I got anybody signed up, and you can raise your hand if you did, but couldn't see a transcription activator presentation. Has anybody done one that wants to say they have? Okay, so I'll go ahead and talk through this, and we'll get to the other stuff. All right, so the transcription activators. So that's what we're seeing here. We learned a little bit about these kind of prematurely when we talked about yeast 2 hybrid assay. That's the funny thing about biology. Sometimes you get things a little different times. But now that we're talking about the DNA elements and regulating transcription, here's a nice picture of that activator that is now closely associated with the whole general transcription, the whole mechanism. So this is kind of cool. Remember that in the yeast 2 hybrid assay, we talked about how this protein has 2 different domains. It has the DNA binding domain and then the activation domain. And we just left it there. We were like, it has there's this thing that the activation domain does, right? But now you can see that we have, once it binds its specific element or DNA element that it recognizes, it's positioned so that the activation domain is now interacting with all of this stuff that regulates what the polymerase is doing, right? So now we've got this binding event, and what happens when proteins bind each other, they can change the shape of each other. So now we've got another element that can, start to regulate whether or not the polymerase is ready to slip away and do its job, or it's ready to be locked in so that it's this, we can grab many different polymerase proteins because we have this cool complex here, right? So you guys were tested on this a little bit, but remember that when we're talking about this DNA binding, at the molecular level, we want to also have a picture of what that is. So we've got these cool motifs that we talked about, the zinc fingers, helix turned helix, helix loop helix, and the leucine zippers. So you want to maybe review what those each are, but this is a neat diagram that shows you how they are associated with the major groove of that DNA, right? Because we're not talking about DNA replication where you open up the bubble and it's like cool, all that easy, you do open up a bubble, but we are talking about regulating, and so there's that initial binding. So how does that happen? How does all that, the activator, the enhancer, how do they bind? And this is that picture that I hope you have in your head. Okay, so this is a neat little fact here. So the activation domains can be there. It's still kind of an open question what they do, or it's not that it's an open question. It's that there are many different jobs that they can do, and when it's spatio-temporal, right? So it's like when is what gene is it working on, and what's going on with the cell at that time, stuff like that is going to change it. But these are these are some of the things that they do. So it does interact with the mediator and the general transcription factor, that's well known. And it can also be involved in modifying the chromatin structure. So it can be involved with the histones as well, okay? So it's a pretty diverse protein in what it does, or domain. All right, so repressors. I love that these are named nicely. So you've got enhancers, you've got the activators. The repressors are the ones that are going to regulate gene transcription by preventing some binding of something and preventing the polymerase from doing the binding and the reading and all of that stuff it's supposed to do. So here's in A, you see a repressor is actually binding the DNA binding domain for an activator. Okay, so that's competitive. If they both can bind the same thing, it looks very similar to that domain. It's like the DNA binding domain of this whole protein, but it's lacking the activator. So it's really like a competitor. This one over here, it actually has a DNA binding domain and a repression domain. So whatever interaction that happens between this part of the protein and this whole complex, it's a negative effect. It's an inhibitory effect. Okay. So maybe it's going to prevent the dynamic change of turning this thing into just first it's a binder, the polymerase, but then it has to become a mover, right? So maybe it's going to stop that change, that dynamic. happens, you know, at the very beginning, the binding of the promoter and, you know, just making that possible. And then there's that change to help it to move. It has to, there are signals within the cell that are going to regulate each one of these steps. Like in biology, at this point, you guys know there's so many multi-step processes, right? And there's, at every step, there's a regulation that can happen. So this diagram shows us some, you know, some of the things that can happen in the, you know, in the next steps, right? So we have our, just to see how it's going, we have the pre-initiation complex, so that's the binding of all of those transcription, the TF2s and the modification of the C-terminal domain. Now there is a position, there's an event where you can pause the polymerase. So the C-terminal domain modification, we talked about that already, that allows it to separate and start to move, right? And it turns it into the elongation version. But there are these other regulatory factors, so more factors that can bind. You've got NELF and DSIF. So I put the long wording on these things. So negative elongation factor is NELF. And I'm not seeing the, well, yeah, so but regardless of what the long name is, these are two transcription factors that will actually bind the polymer, or regulators that will bind the polymerase after it has left, and then they actually stop the motion. So instead of continuing down the line, they will stop that motion right there. Just a, you know, a little bit away from that mediator complex, that big multi-protein complex. When I see this, you know what I think about? This is so silly, I'm such a mom. But it's like when you load all your 4 kids in the car that they all need like 500 things, right? Like the bag, the saxophone, you know, the gym bag, like all the things, their water bottles, you know, their piece of candy for later. Always the random thing. But we have all the things that happen, that's what's happening here. And then you pull out of the driveway and you stop, if you're smart, and you say, what have you forgotten? And somebody always forgets something. So that's what I think about with this. Like there is a moment where there's just like, okay, are we actually ready? Are we actually ready to do this? And that's what NELF and DSIF, they're the reminders. They're the reminders for that. Okay, so the paused polymerase, then in order to unpause it, we have phosphorylation of these factors. So we've got we remove NELF and PTEFB binds. That's positive transcription elongation factor. So negative versus positive. And then that is we got everything we need and we move on. Okay, so remember what phosphorylation does, it changes the shape of those small structures, and then it's like it's a gas pedal or it's an on switch or something like that. Oh, that's it. We just talked about phosphorylation, but we're going to talk about acetylation, methylation, and things like that bind. Just an interesting thing about epigenetics, like just to make it exciting, epigenetics is something that can be different among all of us, and it can be different like depending on what's going on in your life and stuff too. So there was an interesting thing, because of course I'm a mom again, but there was, all of my kids were by C-section, and I read a really interesting article after I went through like the 4th. Don't ever do it, but there is a really significant difference in the epigenetics of babies born in different ways. So that was a really interesting thing too. So it's important. Okay. So and then RNA processing, we had our microRNA presentations and the turnover of RNA and stuff like that. Okay, so I'm gonna actually review this stuff after you guys talk. So my first presentation, let me just think about this. Hold on. Escape. first one's gonna be histone modification, and I have a couple of them. Can I get this group to come on down? I love the colors in this photo. So right, left, forward, back.
So we had histone modification.
So before we begin, histones are highly basic proteins that function to compress DNA within the nucleus to form chromatin, which provides a platform for regulating gene transcription. And those are the histone proteins, and then those are some of the major enzymes.
Okay, so the main role for histone proteins is to bind into DNA. So different types of histone proteins come together to create a nucleus complex, and the DNA is wrapped around that nucleus into a condensed form known as the chromatin. Histone proteins also play a role in gene expression, in controlling gene expression. And then for the enzymes, they play a role in covalent modification by catalyzing the additional removal of chemical groups. So the two most common examples of covalent modifications are acetylation and phosphorylation.
Some of the contributions to the gene regulation are core histones, which package DNA molecules into linker histone, and then enzymes, there's coactivator and co-repressor, and the coactivator increases the accessibility factors that promote transcription, and then co-repressor removes acetyl groups on histones to counteract the effects of the HAT.
Post-transcriptional modification can also take place where it alters the chromatin structures and recruits histone modifiers. Examples of post-transcriptional modifications include acetylation, where is a process of adding an acetyl group to the lysine or adenine residues on the internal tail of DNA. And then methylation can also take place where is the additional methyl group, and acetylation increases the overall rate of gene expression, while methylation represses the overall gene expression. Yeah, so.
Thank you. Pictures again. So the remember what the chromatin is, you know, this is how we package the DNA, and before we can really get anything started, we have to unpackage it, right? So here is that picture. I know that these guys really nicely listed off, so they had that beautiful slide. Use that slide, and I will get those things posted soon. But they listed off the proteins. You know, we have the H1, and that's a cool picture because it's showing it kind of like a clip. But we've got that, these are basic, they said these are basic proteins, and remember DNA has kind of got that negative charge on it, that phosphate. So it wraps around this core, and it's these 8 proteins that are together. So here is our group just mentioned that there's some chemical groups that are added to the DNA to change the way this thing is packaged, right? So here we have the amino terminal tail domain of the histone protein. This is actually the histone protein just linearized, the amino acid sequence. So here you can see that at position 9, 14, 18, 23, those lysines, right? They are acetylated sometimes. Okay, so acetylation of the amino terminal domain is adding this acetyl group to that position. So looking at that chemical structure, you can see there's a great big functional group that changes that. So HATs, add it, HDACs, remove it. So here's those proteins at work. They bind that structure and they make that modification. All right, so you can get these very specific epigenetic gene patterns that regulate this histone packaging. And here we have, this is the active chromatin, and notice we have, we have methylation, acetylation, phosphorylation even of, 4, 9, 10, 14, 17, 18, 23, and 26. That's active. So just the way I think about this is when you're looking at the active one, just the simple, what you're seeing there's a lot of these extra functional groups added on there. And you're making it bulky, you're changing the structure of the histone protein. So are they going to be able to package in that nice core? It's gonna change the way they package and they bind things. So here's the inactive chromatin, so this is stuff that we're not transcribing, and there's some methylation, but there's not a whole lot of stuff on there. Okay, so we got that nice tight binding of the core together. And the H1 is kind of clipped it off. Okay, so we saw our enhancers, so the promoters and enhancers, they will have a distinct chromatin feature. Okay, so they actually will be free of nucleosomes, those enhancers, because they have to be bound, they have to be available and free so that they can do that activation, the enhancement, the turning on of that stuff. And remember the activation proteins, sometimes the activation domain will come and do the work of changing the histones as well. All right. Let's see, am I going too far? What's our next one? I forget what I had on my thing. Histone modification. All right. Okay, so that chromatin has to be remodeled, and without walking through this exactly, one point that's really important to remember is that that's a dynamic thing. What do I mean by it's dynamic? It means it's going to happen at a certain time. It's something that's ever changing. Okay. So let's see. So the protein complexes that alter contacts between DNA and histones are. What am I trying to say here? I'm so sorry. Hold on. Oh, okay. Oh, okay. So once transcription is initiated, let me just ask really quick, is there anybody that did elongation factors? Did I have that on my list? I don't think I did. Elongation factors. All right. So after that we have transcription initiated, then there are elongation factors that can come in and they can continue to, they're going to help with the continuous processing or movement of the RNA polymerase, and one of those things is to change the chromatin as the polymerase is moving on. Just walking through our diagram here, we've got our nice little beads on the string, our nucleosomes. We have binding of our transcriptional activator and chromatin remodeling factor. So here we have chromatin remodeling factor. And this thing is going to interact with the C-terminal tails of those histones, and it's going to continue making those modifications. So another protein that can make the acetylation, all that stuff. Okay, so HDACs we already talked about are the acetylators, and then we have chromatin remodeling factor that's also going to make chemical modifications. So here we have, notice it's bound to the activator, and this is a very specific activator. Job of the activator is binding that chromatin remodeling factor. This changes the C-terminal domain. We remove that bead, right, and then our polymerase is able to go where it's got to go. Okay. So really common theme here is that C-terminal domain of those histones. So don't forget that C-terminal domain of those histones is where those modifications can happen to pull them up. So epigenetics. This refers to the, where is that methylation going to happen? What type of modifications and what positions? This is actually something that can be inherited. Okay, right, because DNA is inherited, and then and because of that, protein sequences, amino acid sequences are inherited as well. Okay. So in our diagram, we've got our nucleosomes with our modified histones. We have some sort of modification to them, and that's on the parental chromatin. The DNA is replicated, and we get the same because the proteins come from a gene. We get the same thing, but we also have, overall the gene expression, the mechanism of opening and closing genes is going to be inherited too. Here we have, let's see, we have a parental nucleosome distribution, and then we have incorporation of our new nucleosome. So as it's being replicated, we can have more nucleosomes incorporated in the strand too. It's not just every 500 we have a nucleosome or something like that. We can add them as the cells or as the DNA is being replicated. All right, so acetylation we saw a nice picture of already. So here's methylation. So we've got our cytosine, and position number 5 is a great place to add a methyl group, CH3. I know we had to memorize those at one point, right? So CH3 is added there. So here's our methylated parental DNA, and then we have duplication, and we get methylation of the daughter DNAs. I don't know if you've ever learned about this, but we've talked about restriction endonucleases before. I'm kind of bringing something in that's not on the slide, but restriction endonucleases. Remember how they work? They bind the bacteria, produce them so that they can chew up DNA, the foreign DNA that comes in. So one question you might have been asked before is, why do the bacterial enzymes chew up foreign DNA and not chew up their own DNA? It's because this methylation pattern right here is something that can actually, I think my thing finally died. That methylation modification is something that actually can prevent that restriction endonuclease from binding. So this modification on this is changing the footprint of the DNA. It's going to change what is able to bind to the DNA. But we're talking, so this is, right now what we're talking about is another mechanism of epigenetic control. And the methyl groups are typically added to that 5 carbon position of our cytosines. All right. Okay. So, sorry. So there's all these things that can happen to the protein, to the DNA, and the proteins that are regulating the DNA to tell us what they can do. So it's not just the acetylation of the protein, but there's also methylation of the DNA itself. All right, so I don't know what I'm trying to say here. I'm actually, expression of some genes depends on whether they come from the mother or the father. Oh, okay. Okay, so that methylation pattern of the DNA, that's going to determine which one of your alleles is going to be expressed. Okay, so remember that every single cell has 2 copies of the chromosomes, right? So we have a parental source and a maternal source. And methylation is something that can regulate which one is active. Okay, because we don't have both copies open expressing all the time. So methylation is one way to determine that. So that's some genomic imprinting. So this one here, we have the sperm is carrying a methylated version, and then when the egg and sperm come together, it's the egg's H19 gene that is expressed rather than the sperm's H19. All right, so another thing that is going to regulate these is some of our long non-coding RNAs. So remember, we had a fair amount of our genome that transcribed RNA that had its own job. So here's one of those examples of that single, they have their own job. So we have PRC2 here that is a protein that is carrying a long non-coding RNA. And the key here is that the long non-coding RNA has this complementary sequence or a binding, an ability to bind a certain position, okay? So this is a neat thing. The PRC2 is a protein. It can only bind the DNA by the help of the long non-coding RNA, and it's a methylator. But in this case, what we're looking at is methylation of the histone C-terminal tails also, okay? So another way to regulate whether or not we're opening these things up or not. All right, microRNA, or not micro, but RNA processing. So let's go ahead, it's not really in the, I'm not matching up our exercise really well to our presentation, but let's go ahead and have this presentation on microRNA. And we'll end there, and then we'll come back and finish up on Friday with the rest of this. So it's one of our star groups. And is this okay? My laser's not working, but my advancing is working on here.
That's what we want to hear.
And if it goes out, I can push the buttons. Just let me know.
Ladies and gentlemen, prepare to be dazzled. Today I have a special treat for you. We're going to do an in-depth synopsis over what microRNA is and how it works in the cells. So let's start. What is microRNA? You guys haven't taken biology ever before, so I'll lay it down for you. So they're small, single-stranded, non-coding RNA. A little bit different than the long non-coding that we just talked about. They usually contain about 21 to 23 nucleotides, and then they can bind to the messenger RNA. So what do they do in the cell? They're mostly regulatory RNA, so they control what and the amount of proteins that are made. How it does this is it interacts with the 3' untranslated region of target messenger RNAs to include degradation and translational repression. So let's talk about the molecular players. We got B and T lymphocytes, we got dendritic cells, we got macrophages, and then some other ones that we aren't going to go in depth today about. How it's created. It's first transcribed from RNA polymerases, both 2 and 3. Then it's the generation of precursors, and then these precursors undergo the cleavage to form mature microRNA. First, we're going to talk about T lymphocyte and B lymphocyte. So right below those, you'll see what those actually are in the body if you aren't aware of those. And the T lymphocyte, what it does is it constrains the function of the microRNA based on the T cell erector function. And then the B lymphocyte, the microRNA helps regulate the terminal for the B cell differentiation and function. Now we're going to go into dendritic cells and macrophages. As before, there's a reminder of what those are. Micro-mRNA are important post-transcriptional regulators. So this is after transcription takes place. It's going to be a regulator RNA. And they modulate physiological and pathological processes. MicroRNA in macrophages influences the activation by regulating the multiple gene sets. MicroRNA's role in gene expression and regulation. So, as we talked about, they're the regulatory RNA. So they're going to regulate post-transcriptionally. They're going to bind with the messenger RNA to do this, usually in the cytoplasm. And once that binding occurs, it can either be destroyed or altered. Now, the messenger RNAs are chosen based on their availability and abundance within the cell. And they can inhibit one specific mRNA target expression and later regulate multiple mRNA expressions. Thank you.
Thank you. Okay, what I really liked about this presentation was putting this into perspective. So remember that microRNA, so we'll come back on Friday, we'll talk about processing of just message RNA, but microRNA is what we're gonna do. So once we get DNA transcribed, then we can still shut it down. We can still shut down gene expression. That's what microRNA can do. And I love this part I love this part of the presentation because it puts it into perspective, like in a whole cell. What is the major effect that happens by regulating these RNAs, and it's specific for like these white blood cells. But remember that microRNA is active in all of your cells, and it's going to regulate what the cells are doing because it's going to up or down regulate the expression of something, the function. All right, that's it.